The scientific method involves a series of steps that are used to investigate a natural occurrence. The first step is to identify the problem or question that will be investigated. A scientist will begin a scientific investigation in the library, reading articles written on the topic by other scientists. He or she will also make their own observations and log them into a notebook. This must be done without bias. The opinion of the scientist must not influence the observations made. The researcher will form one or several hypotheses that may explain the observations collected. The hypothesis is a testable statement, a possible explanation for the observations made previously. Experiments are designed to test each hypothesis made for the investigation. Experiments generally are constructed to test only one hypothesis at a time and will include a dependent variable. And an independent variable. The independent variable is a variable controlled by the scientist. The dependent variable is the one that's observed by the scientist. For example, if the experiment is to test the hypothesis that plants grow taller when exposed to light, the amount of light given to the plants would be the independent variable. The scientist can control how much or how little light each plant will receive. The height of the plant is the dependent variable. The hypothesis assumes that the height of the plant will depend on the amount of light that it receives. When this data is represented on a graph, it is customary to have the independent variable as the x-axis and the dependent variable on the y-axis. When designing an experiment, experimental groups may be formed. Control groups do not receive the experimental treatment. Experimental groups, or treatment groups, do receive the experimental treatment. In this case, sunlight. The experimental group is expected to be affected by the treatment. The control group allows the data to be compared to the experimental group to see if there was an effect. However, if the control group were to grow just as well, it could indicate that the experiment was set up incorrectly, or that the hypothesis has to be revisited. Once the experimental design is finalized, the experiments can be run. The data is collected and the results will be analyzed. After the data has been collected and analyzed, conclusions will be made about the hypothesis. If the data supports the hypothesis will be accepted. If not, the hypothesis will be rejected. And then finally, your conclusions will be communicated to your peers. This can be done by publishing a paper. Or you could create a poster for display at a scientific meeting. Or you could communicate your results to your peers in the form of a presentation. Expect to be questioned and challenged, and be open to that criticism. New hypotheses, experiments, and conclusions may lead to looking at the problem in a different way. Today, we talked about the scientific method. Observe and research the problem. Make observations and research your topic of interest. Formulate a hypothesis. Predict a possible answer to the problem or question. Experiment. Develop and follow a procedure. Collect and analyze results. Modify the procedure if needed. And communicate the results. Be prepared to present the project to an audience. Expect questions from the audience. The next slide is a Bart Simpson scenario. Read it carefully and see whether you can answer the associated questions. Bart Simpson believes that mice exposed to microwaves will become extra strong. Maybe he's been reading too much Radioactive Man. He decides to perform this experiment by placing 10 mice in a microwave for 10 seconds. Do not try this at home. Bart is a misguided fictional character. He compared these 10 mice to another 10 mice that had not been exposed. His test consisted of a heavy block of wood that blocked the mouse food. 8 out of the 10 microwave mice were able to push the block away. 7 out of the 10 non-microwave mice were able to do the same. What was Bart's hypothesis? Bart's prediction, or his hypothesis, was that mice exposed to microwaves will be extra strong. Identify the control group. The mice that were not exposed to the microwaves are the control group. Identify the independent and dependent variables. The independent variable in this experiment, the variable that can be controlled by the scientist, would be the microwaves. The dependent variable in this experiment would be the strength of the mice. The strength of the mice would be observed by the scientist. What might be some of the possible constants? It is very important that any factor that's not a part of the hypothesis not change during the experiment. For example, the same block of wood would be used for all experiments to make sure that didn't become a variable. The same amount of food and the same type of food would be fed to all the mice. The experiment would be run at the same time of day so that all the mice would be equally rested. And all the mice in the experiment would be about the same size. There's probably other constants that you can think of, but these are some examples. What should Bart's conclusion be? Because there was really no difference between the experimental group and the control group, Bart should conclude that microwave exposure does not significantly change the strength of the mice. Okay, let's get this information down into our notes.